The Pharmacovigilance Risk Assessment Committee, PROC, that I chair has looked in great detail at the cases of blood clots reported after vaccination with COVID-19 vaccine AstraZeneca. As soon as these concerns arose last week, the committee started a comprehensive review of all the available evidence and working around the clock, making use of all the resources at our disposal. I want to thank the colleagues from the Belgium team that have led this assessment and worked side by side with colleagues from national competent authorities and the European Medicines Agency, as well as scientific experts in blood disorders, who we asked to provide advice in the assessment. The PROC has come to the following conclusions. This vaccine is safe and effective in preventing COVID-19, and its benefits continue to be far greater than its risks. PROC has found no evidence of a quality or a batch issue. The committee has assessed and discussed preclinical and clinical trial data, published literature, and adverse events related to the blood clots that have occurred during the vaccination campaign, reported by vaccinees and healthcare professionals. Critical to the review has been the careful scientific assessment and clinical scrutiny of the individual cases from the different member states. PROC noted that the number of thromboembolic events reported after vaccination is lower than the expected in the general population, and PROC has concluded that there is no increase in the overall risk of blood clots with this vaccine. Moreover, because the vaccine is effective in preventing COVID-19 disease, which in itself is a cause of blood clots, it likely reduces the risks of thrombotic events overall. However, there are also still some uncertainties. We have seen some very rare case reports describing specific unusual events of a combination of thrombosis and thrombocytopenia and bleeding. In a few cases, tiny clots developed in multiple blood vessels in the first 7 to 14 days after vaccination, a condition called disseminated intravascular coagulation. Also, in a few cases, we saw clots developing in blood vessels draining blood from the brain, and this condition is known as a cerebral venous sinus thrombosis. These conditions are linked to low levels of blood platelets, also known as thrombocytopenia, and the evidence we have is at the moment not sufficient to conclude with certainty whether these adverse events are indeed caused by the vaccine or not. So PROC will continue to gather more information on these conditions, including additional studies, and PROC will keep them under very close monitoring. PROC also concluded that it's very important that these events are rare. As of last night, 7 cases of this disseminated intravascular coagulation and 18 cases of cerebral venous sinus thrombosis were reported out of almost 20 million vaccinated people. Furthermore, such cases were not identified in the large clinical trials, but that highlights the importance of being vigilant once the vaccine is administered to large populations. PROC felt also that it is of great importance that healthcare professionals and people coming for a vaccination are aware of these possible adverse events and that they can spot possible signs or symptoms that may occur after the vaccination. Therefore, PROC has recommended to add a warning to the SMPC and the patient leaflet and a description of these cases, so that this information should be provided to healthcare professionals and to the public. EMA and national health authorities will ensure that healthcare professionals and the general public are made aware of this information and made aware of the signs and symptoms of concern so that timely action can be taken to mitigate these risks.